Hey champs of Examsnet, welcome to the series of chapter-wise discussion. We are back with another chapter, that is photosynthesis, which is part of the plant physiology. Okay, so we have started the plant physiology part. In this part, we will be discussing the chapters photosynthesis, respiration, and plant growth and regulators. Okay, so let's start with this chapter. Here, let's understand the meaning of the term photosynthesis. As we can see that this word consists of two different words, photo and synthesis. Okay, what do we mean by photo? Photo means light, and by light we mean the natural light, that is the sunlight. We also know that the sunlight is the ultimate source of energy, right? Then the other word is synthesis. What do we mean by synthesis? Synthesis means formation or combination. So when we combine different molecules to form a bigger molecule, it is known as synthesis. Okay. So here we are talking about the synthesis or the formation of glucose molecule. Glucose, because the food that is prepared by the plant is glucose, and then this glucose will be stored in the form of starch. Okay, in case of plants, the glucose is stored in the form of starch, whereas in animals the glucose is stored in the form of glycogen. Okay, so here we are talking about the plants, so it will be stored in the form of starch, right? So you understood the meaning of the term photosynthesis. Now, what exactly happens during the photosynthesis? As we saw that sunlight is essential. Okay, so first and foremost, the sunlight has to be captured by the plants. Okay, so the process is not possible if the sunlight is not captured by the plant. So can we make our own food just like the plants? No, right? Because we cannot capture the sunlight. Why we cannot capture the sunlight? Because we don't have special pigments which the plants have. And what are these pigments? The chlorophyll pigments, right? So the plants have chlorophyll in the green parts of their body. Okay, chlorophyll. So chlorophyll is also essential for the photosynthesis to take place, because it is chlorophyll that will help in capturing the sunlight. Okay. So this sunlight will be captured by the autotrophs. Autotrophs like plants. They can be plants, they can be bacteria, cyanobacteria, chrysophytes, etc. Okay. So this sunlight will be captured by the plants to form the glucose molecule. Now we know that the herbivores eat the autotrophs. Okay, autotrophs can synthesize their own food, but herbivores and carnivores cannot synthesize their own food, so they depend on the autotrophs for food. Okay, herbivores are those animals which eat plants, right, or which eat herbs. For example, deer. So deer is a herbivore which can eat the autotroph directly or plants directly. Okay, then the carnivores cannot eat the plant directly, but they indirectly depend on the plants because they eat the herbivores. Okay, so carnivores and herbivores both depend on the autotrophs. Herbivores depend directly, but carnivores depend indirectly. Okay. So you must be knowing these things already, because this is very basic thing which we have been studying since childhood, right? Now here we have the equation for the photosynthesis. In this equation you can see we have 6 carbon dioxide molecules plus 6 water molecules. Then we have glucose molecule that is formed as a result of the photosynthesis, and oxygen has been evolved. Okay, so this is the basic equation of the photosynthesis. Now what can we derive from the given equation? First of all, it is a physico-chemical reaction. Physico-chemical reaction means it is a physical reaction as well as chemical reaction. Why do we say that it is a physical reaction? Because sunlight is being used, and sunlight will have photons, right? So photons of sunlight will be used, therefore this is physical reaction. Then chemical energy is formed. Okay, the light energy is converted into the chemical energy because a chemical is formed. Glucose is a chemical, right? We know that the light, the energy cannot be created nor be destroyed. It can only be changed from one form to another. So in this reaction, what is happening? Light energy is being converted to the chemical energy. Okay, because glucose, that is a chemical, is being formed. Okay, so this is also a chemical reaction. This is also a chemical reaction. So it is physical reaction as well as chemical reaction. So we can say that it is a physico-chemical reaction. Okay, you understood the meaning of the word physico-chemical reaction, physical reaction as well as the chemical reaction. Second point is that it is an anabolic reaction. Now we know that there are two types of metabolic reactions, anabolic reactions and catabolic reactions, right? Anabolic and catabolic. In case of anabolic, what happens? Smaller molecules combine to form a larger molecule. In case of catabolic reactions, opposite will happen. That means a bigger molecule will be broken down into smaller molecules. But here you can see in the equation that smaller molecules of carbon dioxides are being combined to form the bigger molecule, that is the glucose molecule, right? So this is an anabolic reaction. And when the smaller molecules are combined, then energy has to be utilized. Okay, energy is required. So it is an endothermic reaction as well. On the other hand, in case of catabolic reaction, the energy is released, so that will be the exothermic reaction. But this reaction, the photosynthetic reaction, is endothermic because smaller molecules are being combined to form a bigger molecule which will require energy. Therefore, it is an endothermic reaction as well. Got it? Now the third important point is it is a redox reaction. Now what do we mean by redox reaction? It means it will be reduction as well as oxidation. So both reduction and oxidation will take place in this reaction. Let's see how. We have carbon dioxide initially, right? In carbon dioxide you can see that we have 2 oxygens per carbon molecule, per carbon atom. Okay, so carbon dioxide has 1 carbon and 2 oxygen. That means the number of oxygen is double the number of carbon, right? But when it is converted into glucose, then in glucose you can see we have C6H12O6. That means 6 carbons and 6 oxygen. So here the number of oxygen has become equal to the number of carbon. So we can say that the number of oxygen has reduced. Okay, so there is reduction in the number of oxygen, therefore it is a reduction reaction. Now what happened to water? See, water is the molecule that leads to the evolution of the oxygen, right? So this oxygen that is evolved or released, it is from water, not from the carbon dioxide. We will discuss this later as well. Here you can see that the hydrogen of water has been removed. Okay, hydrogen has been removed from it and oxygen has been released. So removal of hydrogen means oxidation, right? So it is also an oxidation reduction. Okay, from carbon dioxide molecule, the number of oxygen has been reduced, so it is a reduction reaction. Then from water molecule, hydrogen has been removed, so it is an oxidation reaction as well. So overall, this is a redox reaction. Got it? So these were some important points about the photosynthesis. Now one more thing is that we can see that initially carbon dioxides were moving freely in the atmosphere, right? But now when they are fixed, when they are captured and they are fixed in the form of glucose molecule, then they are not free. So we can say that this is also a carbon fixation reaction. Okay, this is also a carbon fixation reaction. So carbon fixation is an important part of this chapter that you have to remember. Okay. Now let's discuss some simple experiments that you would have performed in the earlier classes. These experiments show the importance of sunlight and carbon dioxide. Okay. So the first experiment is the variegated leaf experiment. This experiment shows the importance of sunlight for photosynthesis, right? We already know that the sunlight is essential for photosynthesis, but how this was checked, how it was provided as evidence. So this was the experiment which was useful in proving that the sunlight is important for photosynthesis. So in this experiment, what is done? We take 2 leaves. Okay, this is leaf 1, this is leaf 2. Now the first leaf is totally exposed to the sunlight. Totally exposed to the sunlight. This is the sun. Okay. So the sunlight is coming directly to this leaf. When the sunlight is coming directly to the leaf, what will happen? photosynthesis will take place, and when photosynthesis will take place, then glucose formation will take place. Okay, so the glucose will be formed and it will be stored in the form of starch. Okay, so the formation of glucose will take place, glucose, and then this glucose will be stored in the form of starch, right? Now, how can we check whether starch is formed or not? So we use the iodine test for this. Iodine test. What happens in iodine test? We put few drops of iodines on the leaves or anything that you want to check whether starch is present or not. So you have to put few drops of iodine. If the starch will be present, then this iodine will react with the starch and it will result in the blue-black color. Okay, so iodine will react with the starch and it will give blue-black color. Blue-black color. So if you see blue-black color, then it means that the starch is present, right? Now, in the second leaf, what was done? It was covered, okay, it was covered totally by black paper or anything that will not allow the sunlight to come in contact with the leaf. Okay, so the first leaf was totally exposed to the sunlight, but the second leaf was not exposed to the sunlight. It was totally covered, and when it was covered, then the sunlight could not reach to this leaf directly. Okay, it was blocked by that cover or it was blocked by that black paper. Okay, so since there was no contact between the sunlight and the leaf, no photosynthesis took place. Okay, so photosynthesis did not take place. That means glucose formation also did not take place, and there was no starch stored in this leaf, that is in the second leaf. Okay, so when iodine test was performed in this leaf, then no blue black color was seen because there was no starch present in this leaf, so iodine did not react with the starch to give the blue black color. Okay, so in the first leaf, blue black color was observed by the iodine test, but in the second leaf, blue black color was not observed, which shows that the second leaf does not have starch stored in it. So what do we conclude from this reaction, from this experiment? We conclude that the sunlight is important for photosynthesis to take place. Okay, it was the sunlight which came in contact with the first leaf which led to the formation of the glucose molecule. In the second leaf, there was no contact with the sunlight, therefore no glucose formation take place. Okay, so the sunlight is important for the photosynthesis. Now we have another experiment which shows the importance of carbon dioxide. See, we already know that the sunlight is important. We already know that the carbon dioxide is important. But these are the experiments which prove this statement. Sunlight is important and carbon dioxide is important. For carbon dioxide, we use the moles half-leaf experiment. Half-leaf experiment means we have a leaf. Only half of the leaf is used for this. Okay. So you can see this figure. We have a plant. One leaf is used for this, right? We have a bottle or a test tube. Under this bottle, inside this bottle, half portion of the leaf has been kept. Okay, and what is the importance of this bottle? In this bottle, we have a cotton. We have kept a cotton that is soaked with the with the potassium hydroxide. Okay, so the cotton is soaked with potassium hydroxide. Now, what will happen when potassium hydroxide is present inside the bottle? We know that the potassium hydroxide absorbs the carbon dioxide. Okay, so this potassium hydroxide will absorb the carbon dioxide. So what will happen? All the carbon dioxide that is present inside the bottle will be absorbed by this cotton that is soaked in potassium hydroxide. So the leaf part that is present inside the bottle will not receive any carbon dioxide. Okay, carbon dioxide will not be available to this half part of the leaf, this front part of the leaf. Okay, here carbon dioxide is not available. Whereas the other half part of the leaf, which was kept outside the bottle, was in touch with the environment. right, with the atmosphere, environment or atmosphere, where carbon dioxide was available. So this part was in contact with the carbon dioxide. Okay, so carbon dioxide was available to the other half part of the leaf. Now, it was checked which part of the leaf has starch stored in it, okay, by using the same iodine test. So when the drops of iodine were put on the first half part of the leaf, which was kept under the bottle, then no blue-black color was seen. That means no starch was formed. No starch was stored in this part. Why? Because all the carbon dioxide has been absorbed by the potassium hydroxide inside the bottle. Okay, so the carbon dioxide was not available to this part, so no photosynthesis took place. Right, so no glucose formation took place, whereas the other half part of the leaf, which was exposed to the atmosphere, that part received the carbon dioxide, so photosynthesis was done. Okay, glucose formation took place. Right, so this experiment showed the importance of carbon dioxide. Now we will also discuss some early experiments which were performed by the scientists to come to the conclusions which we already know now. Okay, so by now everything is clear about photosynthesis, but earlier these things were not clear. So various experiments were performed by our scientists to find out these facts about the photosynthesis. Okay, so the first experiment that we have to discuss here is Priestley's experiment, okay, which is also known as Priestley's bell jar experiment. Bell jar, because a bell jar was used in this experiment. Now, Priestley was the scientist who discovered oxygen in the year 1774. So this experiment, which is performed by the Priestley, is also related to the oxygen. Okay, so what was done in this experiment? A closed bell jar was taken. Okay, bell jar like this. We will see the figure. This is a bell jar. Then inside the bell jar, a mouse was kept. Okay, then what did he saw? What did he see? He saw that the mouse was able to survive for some time inside that jar, but after some time it started getting suffocated. Okay, and eventually it died. Okay, so keeping the mouse in the closed bell jar for a long period of time led to the death of the mouse. Okay, so the mouse eventually died. That means there was something inside the bell jar that was utilized by the mouse initially, because initially it survived well, but after some time this thing was consumed. Okay, so the mouse died. Now after that, a burning candle was also kept along with a mouse inside the closed bell jar. So this time also, after some time, the mouse died and also the candle extinguished. Okay, the candle also got extinguished. That means there is definitely something which is being consumed by the mouse and the burning candle as well. Okay, so both are utilizing or using something which is present inside the bell jar, which is not visible to us. Obviously, it was oxygen, but it was not known till now. Okay, it was later discovered that it was oxygen. So here you can see in the first figure, a mouse is kept and a burning candle is kept. After some time, the mouse died and the burning candle also got extinguished. Okay, because all the oxygen were consumed. After this, what was done? The candle was done. Candle was kept, burning candle. The mouse was kept, and in addition, a mint plant was also kept. Mint plant, because mint plant is small in size, so a mint plant was also kept along with the mouse and the burning candle. This time, surprisingly, neither the candle got extinguished nor the mouse died. Okay, and the system remained in the equilibrium. So there was no harm to the mouse or the burning candle when the plant was kept along with them. That means the thing which was being consumed by the burning candle or the mouse was replenished or restored by the plant. Okay. So have a look at this figure. You can see mouse is there, burning candle, and the plant is also there. And after some time, all are fine. Okay, the mouse is also alive, the burning candle is still burning, and the plant is also good. Okay, that means the plant balanced the thing very well. Okay, So what was the conclusion of this experiment? The conclusion was that the plants restore to the air whatever breathing animals and burning candles removed. Okay, so it was later found to be oxygen in the year 1774. Okay, so it was oxygen that was used by the burning candle as well as breathing animal, but this oxygen was restored when the plant was also kept in the bell jar. Okay, so this was an important experiment, Priestley's bell jar experiment. Now the second important experiment was Jan Ingenhousz experiment. So in this experiment, what happened? Hydrilla plant was kept in light and then the same plant was kept in dark. Okay, hydrilla is an aquatic plant, right? Aquatic plant. So when this plant was kept in light, then oxygen bubbles were observed on the plants. Okay, oxygen bubbles were seen. And we know that the oxygens are released during the photosynthesis. We have seen the equation of photosynthesis. We know the photosynthesis reaction results in the formation or the release of oxygen molecules. Okay, so oxygen bubble means photosynthesis has taken place. Photosynthesis has taken place, therefore oxygen has been released. When the same plant was kept in dark, then no oxygen bubble was seen. And no oxygen bubble, that means there is no release of oxygen. So no photosynthesis has taken place. No photosynthesis. So again, this experiment also shows that the sunlight is important for photosynthesis to take place. Okay, when the sunlight was available, then oxygen was released. That means photosynthesis took place. When the sunlight was not available, then there is no release of oxygen. That means no photosynthesis took place. Okay, so again, this shows the importance of sunlight for photosynthesis. So the conclusion is that sunlight is essential for the plant process that purifies the air formed by breathing animals and burning candles. So before this experiment, we saw the Priestley's experiment. We saw in that experiment that the plant purifies the air which was formed by the breathing animal and the burning candle. And for this process, sunlight is important. It was shown by Jan Ingenhousz experiment. Okay. Now we come to the third experiment, Julius von Sachs experiment. So what was the importance of this experiment, Julius von Sachs experiment? This experiment provided the evidence for the production of glucose in the year 1854, and it shows that the green substances were located in the special bodies within the plant cells. So we already knew that the plants have some green substances which are important for photosynthesis. Okay, we don't have those green substances. These are nothing but the chlorophyll. Okay, chlorophyll. In this experiment, Julius von Sachs experiment, it was shown that these green substances, that means the chlorophyll, were located in special bodies known as chloroplast. Okay, so this experiment is actually related to the chloroplast. It shows that the chlorophylls are stored in the special bodies. Later it was named as chloroplast. Okay, during this time chloroplast name was not given. Later it was given the name chloroplast. Okay, till now we only know that there are some special bodies in which the green substances are stored. Later on the green substances were known as chlorophyll and these special bodies were known as chloroplast. So it was shown that the glucose was formed in the special bodies, that is chloroplast, and there it was stored in the form of starch. Okay. So this you have to remember, all the experiments, the conclusion of the experiments. Okay. Now we come to the next experiment, that is T.W. Engelmann's experiment. So this is a really interesting experiment, T.W. Engelmann's experiment. Engelmann made the first action spectra of photosynthesis. What do we mean by action spectra? See, spectra means graph and action means process. So here we are talking about the process of photosynthesis, right? Photosynthesis. So action spectra will show you where maximum photosynthesis is taking place, where minimum photosynthesis is taking place, right? For example, if we take this graph, there are two peaks in this graph. This is peak A, this is peak B. Then here we have very small peak. This is peak C. So out of these three peaks, where maximum photosynthesis is taking place? At the first point, point A. Here maximum photosynthesis is taking place. At point C, minimum photosynthesis is taking place. So the graph that explains the process of photosynthesis is known as action spectra of photosynthesis. Okay. Then there is another type of spectrum that is known as absorption spectrum. Absorption spectrum. So you should know the difference between the action spectra and the absorption spectra. Absorption spectra will show you that how much light is absorbed by a given pigment. Okay, so it will give you the absorption of light. It will show you the absorption of light by different pigments like chlorophyll A, chlorophyll B, xanthophylls, etc. Okay. So in this experiment, Engelmann used a prism to split light into its several components. Okay. We know that when a white light is made to pass through a prism, then it breaks or splits into the 7 different components. For example, this is white light. White light, when it passes through a prism, prism is a triangular or pyramidal shaped structure made up of glass, right? So when a white light passes through the prism, it splits into the 7 colors. What are those colors? These are Vibgyor: violet, indigo, blue, green, yellow, orange and red, right? So this is very basic thing. We all know this. Now when the light was split into the several components, then this light or these colors of light were made to fall on a green alga, that is Cladophora. Okay, so he eliminated a green alga, that is Cladophora, placed in a suspension of aerobic bacteria. Okay, so what happened? A white light was made to pass through the prism. When the white light passed through the prism, it split into the 7 different colors of light: violet, indigo, blue, green, yellow, orange and red. Right? So now different colors of light were available. Now all these lights were made to fall on a green alga, that is Cladophora. So suppose this is Cladophora. This is Cladophora. So different part of the Cladophora received different color of light. This upper part received the violet color light. Then this lower part indigo, then blue, then green, then yellow, orange and then red. Okay, so different part of the Cladophora, this is the Cladophora, was receiving different color of light, right? And we also know that this Cladophora was kept in a suspension of aerobic bacteria. So around this Cladophora, many aerobic bacteria were present, right? What is the work of aerobic bacteria? They consume the oxygen. Okay, so these aerobic bacteria were used to detect the presence of oxygen or release of oxygen. Okay, so these bacteria were the site of oxygen evolution, represented the site of oxygen evolution. How? So out of these 7 colors of light, there were 2 lights where maximum amount of aerobic bacteria were accumulated. Okay, these were blue color light and red color light. Okay, so the aerobic bacteria at other colors were very few. Okay, at your region, at the violet region, very few aerobic bacteria were accumulated. At indigo region, very few bacteria. Green region, again very few. Yellow region, very few. But at blue region, maximum amount of aerobic bacteria were accumulated. At red region also, a large amount of aerobic bacteria were accumulated. So there were only 2 regions where a large number of aerobic bacteria were accumulated. And what do they represent? We know that the aerobic bacteria will get accumulated where maximum amount of oxygen is present. That means these 2 regions were the site of oxygen evolution. Okay, and the oxygen is evolved during the photosynthesis. That means the maximum amount of photosynthesis is taking place in these 2 regions, the region of blue light and the region of green light. Okay, so out of all the 7 colors, which colors were most important? The blue color and the red color, because these were the colors which led to the maximum amount of photosynthesis taking place. Okay, so this is shown in this figure. You can see this is the prism. through which the white light has been made to pass. Then 7 colors of lights are shown here. So you can see that there are 2 regions where maximum amount of bacteria are accumulated, the blue region and the red region. At other regions, very few bacteria, 1 or 2 bacteria were present. So these 2 were the sites where maximum photosynthesis took place. So maximum amount of oxygen were also evolved at these 2 regions. Okay. Now I have already explained the difference between the action spectra and the absorption spectra. So here you can see in this graph we have 3 different graphs. One graph is in dark red color, this one. This is showing the action spectra of photosynthesis, action spectra. Then there are other graphs in the broken lines, in the form of broken lines like this. These are the absorption spectra of different pigments like chlorophyll A, chlorophyll B and beta carotene. So here you can see, I hope you can see this graph clearly. Here we have chlorophyll A. This is the graph for chlorophyll A. This is the absorption spectra of chlorophyll A, which I am drawing again with the yellow color. Chlorophyll A. Right? Then in light green color we have the absorption spectra of chlorophyll B. This one. Let me change the color. I will take blue color now. Okay. So in blue color I am drawing the absorption spectra of chlorophyll B. This is the absorption spectra of chlorophyll B. And then there are some absorption spectra of other pigments like beta carotene. So we don't have to focus on them. They are very different from the absorption spectra of chlorophyll A and chlorophyll B. Okay. The absorption spectra of chlorophyll A and chlorophyll B are similar. Right. Also, we see that the action spectra of the photosynthesis that is shown in dark red color is coinciding or roughly resemble the absorption spectra of the chlorophyll A and chlorophyll B. So there is a relation between the action spectra of photosynthesis and the absorption spectra of chlorophyll A and chlorophyll B. That means which pigments are more important for photosynthesis? Chlorophyll A and chlorophyll B. There are other pigments also like beta carotene, xanthophyll, etc. But the most important pigments are chlorophyll A and chlorophyll B. Because in this graph we can see that the action spectra of the photosynthesis is resembling the absorption spectra of chlorophyll A and chlorophyll B. I hope you understood this. Okay. Should I write this here? Action spectra of photosynthesis roughly resembles the absorption spectra of chlorophyll A and chlorophyll B. Okay. Now the last experiment is Van Niel experiment. Okay. So Van Niel was a microbiologist. He was a microbiologist. So he performed the experiment on microbes or microorganisms. So he researched on purple and green photosynthetic bacteria. Till now, plants were used to study the photosynthesis. But Van Niel experiment, being the microbiologist, experimented on the purple and green photosynthetic bacteria. Now, till now, we know that during the photosynthesis, oxygen is released, right? The photosynthesis leads to the evolution of oxygen. But when the photosynthesis was performed using the purple and green photosynthetic bacteria, then it was observed that oxygen was not released. Instead, sulfur or sulfate were released. Okay, so it was concluded that hydrogen from a suitable hydrogen donor is required to reduce the carbon dioxide. Let's understand this. We know that during the photosynthesis, carbon dioxide is used, then water is used. It leads to the formation of the carbohydrate or glucose, and in addition, oxygen is evolved, right? This was already known. Now here we know that the photosynthesis is a redox reaction. Reduction and oxygen both are taking place. So what is happening to carbon dioxide? Carbon dioxide is getting reduced. How? There are two ways in which the carbon dioxide is getting reduced. First, we know that the oxygen is being removed from carbon, right? Because on the left hand side we have 2 oxygen atoms per carbon atom, right? But on the right hand side in glucose we have 6 carbons and 6 oxygen, right? So the number of oxygen has reduced in the carbon dioxide. So reduction has taken place. Another way in which the carbon dioxide has been reduced is by the addition of the hydrogen. Okay, glucose means C6H12O6. So initially the carbon dioxide did not have any hydrogen, but when this is fixed in the form of glucose, then it has hydrogen as well, right? That means oxygen has been removed from the carbon dioxide and hydrogen has been added to the carbon dioxide. Both mean reduction of the carbon dioxide, okay? The removal of the oxygen also mean the reduction, and the addition of hydrogen also mean reduction. So reduction has taken place. So we can say that the carbon dioxide has been reduced. So hydrogen has been added. From where did this hydrogen come? It came from water in case of photosynthesis of plants, right? But when the photosynthesis was observed in the purple and green bacteria, there it was not water which donated the hydrogen. It was something else. Okay, it was hydrogen sulfide. So, using this experiment, Van Meel concluded that it is not always water. It can be something else like hydrogen sulfide as well. So, he said that it is a hydrogen donor. There will be a hydrogen donor, suitable hydrogen donor, which will donate the hydrogen to the carbon, and the carbon dioxide will get reduced. Okay, so this hydrogen donor can be anything. For example, H2A. It can be H2A. So, if H2A is the hydrogen donor, in that case the hydrogen will be donated to the carbon. Okay, so glucose will be formed and A2 will be released, not oxygen, because now we don't have water, so oxygen will not be will not be released. When it is water, H2O, in that case oxygen will be released, and if it is hydrogen sulfide, in that case sulfur or sulfate will be released, not oxygen. Okay, so by this experiment we understand that it is not always water that donates the hydrogen. It can be hydrogen sulfide as well, depending on the organism to organism. Okay, and it can be anything. It can be other donors also, right? And it is not always oxygen that will be evolved. There can be sulfur also. Okay, depending on what is the hydrogen donor available there. So these were the important experiments. Now we come to the main part of the chapter. Okay. Till now we were discussing the historical background. That means the earlier experiments which led to the current knowledge. Now we will discuss what is the main part of the chapter, photosynthesis. So where does photosynthesis take place? Location of the photosynthesis. So this is a very simple answer. We know that the photosynthesis takes place in the green parts of the plants. For example, leaves, or in the green stems, right? Now even in the leaves there are some special cells known as mesophyll cells, which contain special bodies known as chloroplast, and the chloroplasts have the green substances known as chlorophyll. Okay, so chlorophyll are these are very important for the photosynthesis to take place, right? Therefore, the photosynthesis takes place in the chloroplast of the mesophyll cells. Okay. Also, we know that the chloroplasts generally align themselves along the walls of the mesophyll cells to get the maximum amount of sunlight. Okay. So where does the photosynthesis take place? In the mesophyll cells of the leaves, because the mesophyll cells have the chloroplast, and the chloroplast contains the green substances known as chlorophyll, important for the photosynthesis. Okay. Now here we have the diagram of the chloroplast. In the chapter cell, we have already discussed about the detailed structure of the chloroplast. We know that in a chloroplast we have coins-like structure known as thylakoid. So this is a thylakoid. Okay. Thylakoid. Then these thylakoids are stacked one upon another. just like the piles of coins, and these stacks are known as grana. Okay, so what are grana? Grana are the stacks of the thylakoids. Okay, then there are several such grana present, right? You can see this is one grana in this figure, this is another grana, this is another grana, and also you can see that all these grana are connected together. Okay, so there is a structure which connects the thylakoid of one grana to the thylakoid of another grana. These are known as the thylakoid, the lamellae. Okay, these are known as stoma lamellae. Stoma lamellae. What is the function of stoma lamellae? They connect the thylakoid of one grana to the thylakoid of another grana. Okay, and the matrix is known as stoma. Okay, stoma is the matrix where various enzymes are present. Okay, so some enzymatic reactions take place in the stoma. Now, in the photosynthesis, two types of reactions take place: light reaction and dark reaction. Okay, so these are the most important part of this chapter: the light reaction and the dark reaction. So light reaction is associated with which part of the chloroplast? Grana. Okay, the light reaction takes place in the grana, that means the stacks of the thylakoid, and the dark reactions take place in the matrix of the chloroplast, that is the stoma. So this you have to remember: light reaction takes place in the grana and the dark reaction takes place in the stoma. Now what is the difference between the light reaction and the dark reaction? Light reactions are directly light driven. Okay, they are directly dependent on light, directly depend on light, and they lead to the formation of ATP and NADPH. So light reaction leads to the formation of ATP and NADPH, which are very important. Without the formation of ATP and NADPH, no further reaction can take place. Dark reaction, which takes place in the matrix of the chloroplast, that is stoma, leads to the carbon dioxide fixation. Carbon dioxide fixation in the form of carbohydrate, or glucose or sugar. Okay. Now, this dark reaction requires the ATP and NADPH. Okay, so ATP and NADPH are required during the dark reaction. Right? If ATP and NADPH will not be formed, then dark reaction cannot take place. So dark reactions are also indirectly dependent on the light reaction, because they depend on the products of the light reaction. Okay. So it does not mean that the dark reaction does not need light. It needs light, because if light will not be present, then ATP and NADPH will not be formed. And if ATP and NADPH will not be formed, then dark reaction will also not take place. So dark reaction are also dependent on light, not directly but indirectly. So why they are known as dark reaction? Just to differentiate them from the light reaction. Okay. So what did we see? We saw that the light reaction takes place in the grana and it leads to the formation of ATP and NADPH. Okay. Then the dark reaction takes place in the stoma and it leads to the carbon dioxide fixation in the form of carbohydrates. Now if we write the complete equation of photosynthesis, which we had to discussed earlier, but let's see. The complete reaction of photosynthesis will be 6CO2 plus 12H2O. It gives the glucose C6H12O6 plus 6 molecules of water plus 6 molecules of oxygen. Okay, this is the complete reaction of the photosynthesis. Okay, just go through the book, you will see there. Now, have you seen that the plants are green in color, right? But have you ever observed that not all the plants look very similar? Okay, they are different from each other. Okay, the shades of green vary from one plant to other plants. Some plants are dark green in color, some are light green in color, some are yellowish green, and some might appear to be orangish, right? Why is it so? Because the plants contain different types of pigments. There is not just a single pigment which give color to the plants, right? There are different pigments which provide color to the plants. So these pigments are carotenoid, xanthophylls, chlorophyll A, chlorophyll B. Okay, there are 4 main pigments that are present in the plant. These are the 4 main pigments. Carotenoids give the color yellow to yellow orange. Okay, so if the leaves of the plants appear to be yellow, orange or yellow, you can understand which pigment is responsible for this color. Carotenoid pigment are present, okay, which are also present in the carrot. Okay, carrot also has this pigment which gives the color orange to the carrot. Then there is another pigment called xanthophyll which gives yellow color to the plant. Then chlorophyll A and chlorophyll B. Chlorophyll A gives bright or blue-green color to the plant, whereas chlorophyll B gives yellow-green color. Okay, so the difference in the shades of green are due to the different pigments present in the plants. Okay, we also know that these are the pigments which absorb light, absorb the sunlight during the photosynthesis, right? And because of that, the plants can perform the photosynthesis. We cannot perform the photosynthesis because we do not have these pigments in our body. Okay, so these pigments are very important. If these pigments will not be present in the plants, then they will not be able to absorb the sunlight to perform the photosynthesis. Okay, now we have already discussed the difference between the action spectra of the photosynthesis and the absorption spectra. Okay, absorption spectra means what? Absorption spectra is the graph that shows the absorption of light by different pigments of the plants like chlorophyll A, chlorophyll B, xanthophylls, carotenoids. All these pigments absorb the sunlight. Okay, so they absorb the light of different wavelength. So this absorption of light by the pigments is represented by the absorption spectra. So you can see this graph. Okay, this graph shows the absorption spectra of chlorophyll A, chlorophyll B and the carotenoids. Okay, in blue color, focus on the blue color graph. It shows the absorption spectra of chlorophyll A. Okay, green color graph shows the absorption spectra of chlorophyll B and the red color graph shows the absorption spectra of carotenoids. Okay, on the y-axis, along the y-axis we have the absorption of light. Okay, by the pigments, different pigments, and along the x-axis we have the wavelength of light in nanometer that are absorbed by these pigments. So if you see the absorption spectrum of the chlorophyll A, that is in blue color, you can see the peak. This is one peak and this is another peak. Okay, you can see peak A and peak B. So the first peak is somewhere between 400 and 500 nanometer and it is near to 400. Okay, so the first peak of the absorption spectrum of chlorophyll A is near 400 nanometer and the second peak of the absorption spectrum of chlorophyll A is near 700 nanometer here. Okay, so first one is near the 400 nanometer, which is the region of blue light. Okay, this is the region of blue light. And the second peak is near 700 nanometer, which is the region of red light. Okay, we have also discussed that the maximum amount of photosynthesis takes place in the blue and red region, right? And here we can see that the chlorophyll A is absorbing maximum light of around 400 nanometer and 700 nanometer. That is, it is absorbing maximum amount of blue light and the red light. So can you relate the relate the absorption spectrum of chlorophyll A and the action spectrum of photosynthesis? Both are related. Okay, similarly you can see the absorption spectrum of chlorophyll B, which is shown in green color. So here the first peak is near 500. Okay, which is slightly different from the peak of the chlorophyll A, and the second peak is around 650 nanometer. Okay, here most important one is the chlorophyll A because it is absorbing maximum amount of blue light and red light, which are most important for photosynthesis. Okay, so you have to focus on the chlorophyll A here. Just have a look on the next graph. Here we are comparing the absorption spectrum of the chlorophyll A, that is shown in the blue color. This is the absorption spectrum of chlorophyll A, and the black color graph shows the action spectrum of photosynthesis. Okay, so you can see these two are resembling, these two are coinciding. Okay, the action spectrum of photosynthesis is resembling the absorption spectrum of chlorophyll A. So the peaks of both these graphs are similar, okay, around the same region, that is the blue region and the red region. So we can see now that blue and red region are the most important for photosynthesis. Maximum amount of photosynthesis takes place in the blue region and the red region, and the maximum amount of photosynthesis is performed by which pigment? Chlorophyll A, right? So we can say that the chlorophyll A is the most important pigment among all the pigments of the pigments that are present in the plants. Okay, so different pigments were present in the plants, but most important of them is the chlorophyll A. Okay, so the chlorophyll A is considered to be the chief pigment in the plant. Okay, it is the chief pigment. Chlorophyll A is the chief pigment. But does that mean that other pigments are not important? No. The other pigments are also important, although they are not chief pigment, they are accessory pigments, okay? Accessory pigments. Other pigments are known as accessory pigments. So what is the importance of the other pigments? They absorb light of different wavelengths, okay? Chlorophyll A absorbs light of around 400 nanometer and around 700 nanometer. 700 nanometer. That means the blue light and the red light. There are other color of lights or other wavelengths of light which are absorbed by different pigments. Okay. So if different types of pigments will not be present, then only blue and red light will be absorbed. Other lights will not be absorbed. So different pigments absorb light of different wavelengths. So these accessory pigments absorb light of different wavelengths. Okay. At the same time, they also protect chlorophyll A from photooxidative damage. So accessory pigments are also important. Okay, they are not useless. Now we come to the photosystems. So what is photosystem? Photosystem consists of two parts, reaction center and the light harvesting complex. So there are two parts of the photosystem, the reaction center and the light harvesting complex. The reaction center consists of a single chlorophyll A molecule. We know that the chlorophyll A molecule is the chief pigment, right? So this is chlorophyll A molecule. This is the reaction center. Okay. Then there will be light harvesting complex, which will consist of the accessory pigments, hundreds of pigments as well as proteins. Okay. So there are some other pigments which form the light harvesting complex. Okay, so what is the function of the accessory pigments? They will absorb light of different wavelengths and they will transfer these light to the chlorophyll A. So chlorophyll A is the chief pigment, right? And it is the reaction center. So the most important is chlorophyll A. Other pigments will be helping the chlorophyll A by giving it or transferring it the light of different wavelengths. Okay, so they together are known as photosystems. Parts of photosystems are reaction center, that is chlorophyll A molecule, and then the second part is the light harvesting complex, which consists of the accessory pigments. It can consist of hundred of pigments and proteins. Okay. Now there are two types of photosystems, photosystem 1 and photosystem 2, right? Why they are known as photosystem 1 and photosystem 2? Because photosystem 1 was discovered earlier and photosystem 2 was discovered later. Okay, it does not mean that photosystem 1 will take place first and then photosystem 2 will take place. It is not named in the sequence. Okay, it is named just because of the discovery, sequence of the discovery, not in the sequence in which they function. Okay, in fact, when both PS1 and PS2 are present, photosystem 1 and photosystem 2 are present, then photosystem 2 takes place first and then photosystem 1 takes place. Okay. So the name is given only in the sequence of their discovery, not in the sequence of their action. Okay. So do not get confused. What is the difference between PS1 and PS2? See, the light harvesting complex will be present in both. The only difference is that in case of photosystem 1, the maximum absorption peak will be 700 nanometer, whereas in case of photosystem 2, the maximum absorption peak will be 680 nanometer. Okay, so the chlorophyll A molecule, that is the reaction center of photosystem 1, will absorb the light of 700 nanometer. It will be the maximum absorption peak, whereas the chlorophyll A molecule, which will be present in photosystem 2, will absorb maximum the light of 680 nanometer. Okay, the only difference between the photosystem 1 and photosystem 2, other things will be same. That means the light harvesting complex will be present in both. Both will have the chlorophyll A molecule. The only difference will be in the maximum absorption peak. Okay. Now this light harvesting complex is also known as antenna. That also you have to remember. Here we have the figure showing the light harvesting complex. This is the light harvesting complex. This one molecule is the reaction center. This is the reaction center, chlorophyll A molecule, the chief molecule, chlorophyll A. One is the reaction center, and all the others are the helper molecules. Okay, these are the light harvesting complex. Okay, so light harvesting complex is contains all the molecules other than the chlorophyll A, the reaction center. Okay, and they are together known as light harvesting system. Now this is another figure that will explain this to you. You can see here the pigment molecules and the chlorophyll A molecule are shown in different colors. Okay, in green color there are pigment molecules which will receive the light of different wavelengths. Okay, so different pigment molecules will be receiving light of different wavelengths. These molecules are also known as antenna pigments. Okay, together these molecules are known as light harvesting complex. So light harvesting complex contains the pigment molecules other than the chlorophyll A molecule, Then this is the chlorophyll A molecule, that is the reaction center. Okay, this is the chief pigment out of all the different pigments. Okay, so this is electron donor, chlorophyll A molecule, because when it will receive the sunlight, the electron will get excited and the electron of chlorophyll A molecule will jump into an higher orbit where it will be accepted by an electron acceptor. So electron acceptor that will be available at a higher orbit will catch the electron excited by receiving the sunlight. Okay, therefore the chlorophyll A molecule is also known as electron donor, right? So we have chlorophyll A molecule, which is the electron donor. And we know that the light harvesting complex, that is the pigment molecules other than chlorophyll A molecule, along with the chlorophyll A molecule, they will be known as light harvesting system. Okay, so light harvesting complex plus chlorophyll A molecule will be light harvesting system. Okay. Now another important fact that you have to remember is that the electron of chlorophyll A will get excited only when the chlorophyll A molecule will receive the light of 700 nanometer in case of PS1. Okay, so if it is photosystem 1, then the chlorophyll A molecule has to receive the light of 700 nanometer and then the electron will become excited. In case of PS2, the electron will get excited when the chlorophyll A molecule will receive the light of 680 nanometer. Because we know that in case of PS1, the chlorophyll A molecule has the absorption peak of 700 nanometer, and in case of PS2, the chlorophyll A molecule has an absorption peak of 680 nanometer. Okay, so this is the basic difference that you have to remember. Now we come to the light reaction. That is the most important part, because these photosystems are associated with the light reaction. Okay, that you have to remember. Now we know that the light reaction is important because it results in the formation of ATP and NADPH. ATP and NADPH, these are the products of light reaction. And then the next type of reaction, that is dark reaction, will depend on these products of the light reaction, ATP and NADPH. Okay. So light reaction is also known as photochemical phase because it is directly dependent on the sunlight. Okay, light reaction is directly depends on light or sunlight, whereas the dark reaction does not directly depend, indirectly depends on the light. Okay, because that depends on the products of the light reaction. Then what happens during the light reaction? The first thing is light absorption. Since light reaction, the word light is mentioned in the name, it means light is important. Okay, so the first step of the light reaction is absorption of sunlight. So this is the sunlight. Here we have the photosystems. This is chlorophyll A molecule, and there are other pigment molecules. Okay, so chlorophyll A molecule I have shown in bigger size, and other pigment molecules are in smaller size. So the sunlight will be received by these pigments. Okay, different pigments will receive the light of different wavelengths, but the chlorophyll A molecule will receive the light of 700 nanometer or 680 nanometer. Okay, depending on whether it is PS1 or PS2. In PS1, it will receive the light of 700 nanometer. In PS2, it will receive the light of 680 nanometer. Okay, so the light absorption is the first step. Second is water splitting. Water will split, and another important thing that you have to remember is water splitting is associated with PS2 only. PS2, not PS1. Okay, let's recall one more fact that we have already discussed. We know that PS2 acts first. Okay, it will act first, and then PS1 will act later, when they both are working actively. Okay, when they both are working together, when both PS1 and PS2 are available, in that case, which photosystem will act first? PS2 will act first, and then PS1 will act. Okay, therefore water splitting is associated with PS2, not with PS1. Okay, they are named in the sequence of their discovery, not in the sequence of their action. Then when water will split, oxygen will get released. Okay, water splitting will result in the release of oxygen. Let's write the equation for the release of water. Here I am writing the equation. We have water, H2O. It will split, so it will give 4 protons, then 4 electrons, and then oxygen. So what are the products of splitting of water? Protons, electrons, and oxygen. And we know that oxygen is also one of the net products of photosynthesis. So this is the oxygen that is the net product of the photosynthesis. Okay. Then what happens to the proton that is released during the splitting of water? These protons will get accumulated in the lumen of the thylakoid. Okay. They will accumulate in the lumen of thylakoid. And this is very important fact because we will need to recall this fact in the next topic, chemiosmotic hypothesis. Okay, so you have to remember that the protons that are released during the splitting of water will get accumulated in the lumen of the thylakoid. This will result in the increased concentration of proton in the lumen of the thylakoid. Okay, and then the third product is electron. So this electron will be used during the light reaction. Okay, how this electron will be used during the light reaction, that we will see next. Okay, and we also know that the light reaction results in the formation of high energy rich chemical intermediates, that is ATP and NADPH. Okay, so these are the products of the light reaction. There are involvement of several protein complexes as well. Okay, so the light reaction will also involve protein complexes. This also we will see, and it consists of photosystem 1 and photosystem 2. Okay, so both photosystem 1 and photosystem 2 are parts of the light reaction. Now let's see electron transport. Here we will see how the electron that was released during the splitting of water will be used during the light reaction. Okay, so we know that when both photosystem 1 and photosystem 2 are available, in that case, which photosystem will act first? Photosystem 2. So the process will start with photosystem 2. Okay. Now we will come to this line after looking at the figure. Okay, first let's have a look at the figure, then only you will understand this. You can see first we have photosystem 2 and then we have photosystem 1. Light is absorbed by the chlorophyll A molecule. Here we have the light harvesting complex of the photosystem 2. Here we have the light harvesting complex of photosystem 1. Okay. In blue color, we have the light harvesting complex of photosystem 2. In green color, we have the light harvesting complex of photosystem 1. Light will be absorbed. First, focus on the photosystem 2. Okay. Light is light is absorbed. What will happen when light is absorbed? We know that the chlorophyll A molecule of the photosystem 2 will accept, or it will absorb the light of 680 nanometer. Okay, let me draw this again here. We have the sunlight. We have the light harvesting complex of photosystem 2. Here we have the chlorophyll A molecule. This is the chlorophyll A molecule. This is photosystem 2. Okay. When it will absorb, the chlorophyll A molecule will absorb the light of 680 nanometer, then the electron will become excited, okay, and then this electron will jump into a higher orbit where it will be accepted by the electron acceptor. Okay, electron acceptor will catch this electron which was excited by the absorption of light. Okay, then what will happen to this electron? This electron will be passed to various electron carriers. Okay, so first of all, when the electron jumps into a higher orbit, this is an uphill movement. Okay, uphill movement. What do we mean by uphill? When it moves from a lower redox potential to higher redox potential, it is known as uphill movement. Okay, so this is in terms of the redox potential scale. Okay, so when it is moving from lower redox potential to higher redox potential, this movement is known as uphill movement. So this movement of electron from this to a higher orbit was uphill movement. Now this electron will be passed through various electron carriers. So these are the electron carrier molecules which will consist of cytochromes. Okay. So this movement of electron from one carrier to another carrier of the electron transport system, all the carriers, electron carriers together are known as electron transport system. This you can see in this figure. Okay. So electron transport system will consist of various electron carriers. So electron will pass through these carriers, and this movement of electrons will be downhill movement. Okay, downhill, because this time it is moving from a higher redox potential to lower redox potential. Okay, then after passing through the electron transport system, this electron will be transferred to another photosystem, that is photosystem 1. So the electron started from photosystem 2, it moved to a higher orbit, then it passed through the electron carriers of the electron transport system, and now it is transferred to photosystem 1. Okay. So this electron is now received by photosystem 1. Again from here it will move to a higher orbit. That means the movement will again be uphill movement to a higher redox potential. Another electron acceptor will be there. Again it will be caught by the electron acceptor, and then again it will move downhill. Okay, and this time it will move to NADP plus. NADP plus. When NADP plus will receive the electron, it will get reduced to NADPH and H plus. Okay, so it will result in the reduction of the NADP plus, and it will result in the formation of NADPH. Okay, so I hope you have understood this. Now, very important fact: where are ATP and NADPH formed? ATP is formed during the downhill movement of electron through the electron transport system. Okay, so when the electron was moving through the electron transport system, there ATP was formed, because this movement resulted in the phosphorylation of ADP. Okay, and it resulted in the formation of ATP. And during the second downhill movement of electron, NADPH was formed, because the electron was accepted to the NADP by the NADP plus. Okay. So I hope you have understood this. Now we know that the splitting of water is associated with PS2. Why? Because we know that the PS1 will be getting the electron from PS2. Okay, the electron will start from the PS2 and it will be transferred to PS1. So PS1 will be getting the electron from the PS2. But from where will PS2 get the electron? So it will get the electron by the splitting of water. Okay, so splitting of water will take place. Splitting of water. It will result in the release of electron which will be used by PS2. Then it will be passed to the PS1. Okay. This process is known as non-cyclic photophosphorylation. Non-cyclic photophosphorylation. First, let's understand the meaning. Photophosphorylation. Photophosphorylation. Phosphorylation. Photo means light, because sunlight is important here. Okay, this process cannot take place without the sunlight. So light is important. That is why photo is used. Then phosphorylation means addition of phosphate. Addition of phosphate. So during this process, the ADP gets the inorganic phosphate. Okay, the inorganic phosphate is added to the ADP, which results in the formation of ATP. Therefore, this is known as phosphorylation. Okay, and since light is used, light is important, therefore the process is known as photophosphorylation. And why it is known as non-cyclic photophosphorylation? Because the electron started from PS1 and then it was transferred to PS2, and this electron was then used by the NADP plus. Okay, and the NADP plus was reduced to NADPH and H plus. Okay, this electron did not get cycled back to the PS2. Okay, once the electron was used by the PS2, this was not cycled back to PS2. Then we will have another type of photophosphorylation, that is cyclic photophosphorylation, in which the same electron will come back to the photosystem too. Okay, so this is non-cyclic photophosphorylation. And if we look at this figure carefully, the shape of the movement of electron looks like this. Okay, it is like this, N shaped. But if you look at this from this axis, suppose from here you see this, it will look like Z. Okay, if you look from the axis of the redox potential, the shape will look like Z. Therefore, this is also known as Z scheme. Okay, what is the other name? Z scheme. It looks like this, and if you look from this side, it will look like Z. Therefore, this is known as Z scheme. Okay, so Z scheme is another name for non-cyclic photophosphorylation. Here we will discuss about the splitting of water that is associated with the PS2. Okay, this is associated with PS2. This is important, you have to remember. Splitting of water is associated only with PS2. And PS2 is located in the inner thylakoid membrane. Okay, so this is the equation of the splitting of water. Water molecule splitting resulted in the release of protons, oxygen and electrons. Again we will revise. The protons that are released will get accumulated in the lumen of the thylakoid, which will result in the increased concentration of proton in the thylakoid lumen. Oxygen is one of the net products of the photosynthesis, and the electrons that are released will be used during the light reaction. Okay. This splitting is associated with PS2, we have already discussed. Oxygen is one of the net products of the photosynthesis, and the electrons, these are available to PS2 and they will then get transferred to the PS1. Okay, they will replace the electron removed from PS1. Okay, so we know that from PS2, electron will get excited, it will be accepted by the electron acceptor, then it will move downhill through the electron transport system. Okay, here it will reach PS1, photosystem 1, then again it will move uphill to another electron acceptor, then again downhill to NADP plus, right? And now the electron will be removed. So this electron is eventually removed by the PS1. So to replenish or to replace the electron, we need new electrons, right? And these new electrons will be available by the splitting of water in the PS2. Okay. So this was the non-cyclic photophosphorylation. Now we come to the next type of photophosphorylation, that is cyclic photophosphorylation. So in non-cyclic photophosphorylation, non-cyclic photophosphorylation, both PS2 and PS1 were available. Okay, PS2 and PS1 both were available. But in cyclic photophosphorylation, only PS1 will be available. Okay, only PS1 will be available. So when will cyclic photophosphorylation take place? When only photosystem 1 is available and photosystem 2 is not available. Okay. So the condition for cyclic photophosphorylation is when PS2 is absent. PS2 is absent. Then cyclic photophosphorylation will take place. Another condition for cyclic photophosphorylation is that even if the photosystem 2 is present, the light of 680 nanometer is not present. Okay, if 680 nanometer light is not present, light is not available, then also non-cyclic photophosphorylation cannot take place, only cyclic can take place. Okay, because photosystem 2 requires the light of 680 nanometer. Okay, now here we have the figure for cyclic photophosphorylation. Light is absorbed by the chlorophyll A molecule of photosystem 1. Okay, this is photosystem 1. In cyclic photophosphorylation, only photosystem 1 is present. And we know that the chlorophyll A molecule of photosystem 1 will receive the light of 700 nanometer, not 680 nanometer. Okay, so it requires the light of 700 nanometer. Therefore, it is written P700. Okay, chlorophyll A molecule of photosystem 1 is known as P700 molecule. So the light is received by this molecule. The electron gets excited. It will move to a higher orbit. It will be accepted by the electron acceptor of the photosystem 1. Now we do not have any other photosystem. So the same electron will pass through the electron transport system and eventually reach photosystem 1 again. Okay, it will be cycled back to the photosystem 1 because there is no other photosystem so that it can be transferred. Okay, so it will be cycled back to the same photosystem. Now during the movement of the electron through the electron transport system, again the phosphorylation will take place. That means the addition of inorganic phosphate to the ADP will take place and ATP will be formed. Okay, therefore it is known as cyclic photophosphorylation. Now what's important here, that in cyclic phosphorylation only ATP has been formed. Where is NADPH? NADPH is not formed here. NADPH will be formed in non-cyclic photophosphorylation, not in the cyclic photophosphorylation. Okay. Now one of the possible location of the cyclic photophosphorylation is the stroma lamellae. Stroma lamellae. So in stroma lamellae, cyclic photophosphorylation takes place because the stroma lamellae does not have PS2. Stroma lamellae lacks PS2. Therefore, the non-cyclic photophosphorylation is not possible. Also, the stroma lamellae lacks NADP reductase enzyme. NADP reductase enzyme. And this enzyme is important for the reduction of NADP plus to NADPH and H plus. Okay, so this enzyme is not available in the stroma lamellae. Therefore, only cyclic photophosphorylation can take place. Non-cyclic photophosphorylation cannot take place. Okay, so here only ATP will be formed. NADPH cannot be formed. Now we have another figure. If you want to look at this figure, you can see this is again the figure of the cyclic photophosphorylation. I have added different figures so that you can understand by the figure that you understand well. Okay, so here we have the light, sunlight absorbed by the chlorophyll A molecule. Which wavelength? 700 nanometer. Okay, because here we have only PS1. So the absorption peak will be 700 nanometer. So this light will be absorbed. The electron will get excited. So the electrons got excited, accepted by the electron acceptor. Here we will have the electron acceptor. Then this electron will be passed through the electron transport chain or electron transport system which will consist of several electron carriers. So these oval shaped molecules are the electron carriers. Okay, and then passing through them, the electron will be cycled back to the same photosystem 1. Okay, and during this process ATP will be formed. ATP will be formed because phosphate will get added to the ADP, which will result in the formation of ATP. Okay, this is must. Phosphorylation is must. Therefore, the name phosphorylation has been given. Okay, so the phosphorylation will be common in the cyclic and the non-cyclic one. Therefore, ATP will be formed in both cyclic photophosphorylation as well as non-cyclic photophosphorylation. But NADPH will be formed only during the non-cyclic photophosphorylation. Okay, so that you have to remember. So the amount of ATP will be more than the amount of NADPH. Here we have the location of the PS1 and PS2. PS1 is available on the outer surface of the thylakoid membrane, and this is stroma facing. It will face the photosystem 1 will face the stroma, and photosystem 2 is located on the inner surface of the thylakoid membrane, so it is lumen facing. Okay, the photosystem 2 faces the lumen. So here let's recall one more fact. We know that the photosystem 2, with the photosystem 2, splitting of water is associated. So water will split here. It will result in the formation of protons, electrons and oxygen. Now these protons, where will they get accumulated? They will get accumulated in the lumen of the thylakoid, because photosystem 2 is facing the lumen of the thylakoid. Okay, that is why the protons released during the splitting of water will get accumulated in the lumen of the thylakoid. Got it? Now the next topic is chemi-osmotic hypothesis. Again, one of the most important topics of this chapter. We already know that during the light reaction, ATP and NADPH are formed, right? ATP and NADPH. But how are they formed? This is also important. Okay, we should know how are ATP and NADPH formed during the light reaction. The formation of NADPH is simple. We know that electron from PS2 passes to the electron transport system and then finally to the photosystem 1, right, during the non-cyclic photophosphorylation. Now after passing through photosystem 1, it reaches the NADP plus, okay, and this NADP plus accepts this electron along with protons present in the stroma and get reduced to NADPH and H plus. So this is how NADPH is formed. Got it? Electron passes from PS2 to PS1 and then it eventually reaches to NADP plus, which accept this electron along with protons present in the stroma and then get reduced to NADPH plus H plus. So simply NADPH is formed. Now how is ATP formed? That is important, and this is slightly complicated. Okay, the formation of ATP. So the formation of ATP during the light reaction is explained by the chemi-osmotic hypothesis. Okay, so this hypothesis is going to explain you the formation of ATP during the light reaction. Now have a look at this figure carefully. This is the figure that is given in the NCERT. First of all, a proton gradient has to be formed. Okay, there is a requirement of proton gradient across the thylakoid membrane, thylakoid membrane. So this oval shaped structure that you can see in pink color, this shows you the thylakoid membrane. So this oval shape is thylakoid. So this is thylakoid membrane. As you can see, there is an outer membrane and there is an inner membrane, right? In this membrane, we have photosystem 2, then cytochromes, and then photosystem 1. Okay, so these photosystems are embedded in the membrane of the thylakoid. Okay, now the space inside the thylakoid membrane is known as the lumen of the thylakoid, whereas the space outside the membrane is the stoma, that is the matrix of the chloroplast, right? So the membrane, the space outside the membrane is stoma and the space inside the membrane is the lumen. This is clear? Now, what will happen? First of all, a proton gradient will be developed and then this gradient will be broken down to release the energy. Okay. Now, how the proton gradient will be formed? Gradient means what? Gradient means a difference in the level. Okay, suppose one side we have more protons and the other side we have less protons, it will lead to a difference in the levels of the proton or difference in the concentration of the proton. So this is known as the proton gradient. Got it? So here we have to develop a proton gradient. That means one side we should have more protons and on the other side we should have less protons. Okay, so where we should have more protons? In the lumen of the thylakoid. That means the space inside the thylakoid membrane. So we have to increase the concentration of the proton inside the lumen of the thylakoid. So how will this increase take place? That is important. So there are two reasons how protons concentration will increase inside the lumen. First of all, we know that the water splitting is associated with photosystem 2, right? Splitting of water is associated with photosystem 2. Also, we know that the photosystem 2 is associated with the inner thylakoid membrane, right? Inner thylakoid membrane. That means the splitting of water will take place towards the inner thylakoid membrane, right? And we also know that the splitting of water leads to the release of protons, electrons, and oxygen. Right? These three things are released during the splitting of water. Oxygen will be one of the net products of the photosynthesis. Electron will take part in the electron transport from PS2 to PS1, right? What will happen to the proton? The proton will get accumulated in the lumen of the thylakoid. So the protons which are released during the splitting of water are getting accumulated in the lumen of the thylakoid. So as a result, the concentration of proton is increasing in the lumen of the thylakoid. So this is just one reason why the proton concentration is increasing in the lumen of the thylakoid. There is another reason as well. We know that there are various pigments in the electron transport system which transport the electron from one place to another. Okay. Now these pigments are also the electron carriers, right? These pigments are electron carriers because they carry the electron from one place to other. Now we will need another diagram here. Let's come to the second diagram. Okay, so this diagram clearly shows you the pigments present in the membrane. Okay, so we have the photosystem 2, then cytochrome complex, and then photosystem 1, right? Here we have PQ. PQ is plastoquinone. Plastoquinone. This figure is not given in the NCERT. I have added this figure. Okay, PQ stands for plastoquinone. PC is plastocyanin. Plastocyanin. Now these are nothing but the pigments or the electron carriers. Okay, they are electron carriers. They will help in carrying the electron from PS2 to PS1. Okay. So they are part of the electron transport chain or electron transport system. Okay. Now here you can see the location of PQ and PC. Plastoquinone is located towards the outer membrane. Okay, we have double membrane. So plastoquinone is located towards the outer membrane on the upper side and plastocyanin is located on the inner membrane. You can see the difference in their location. Plastoquinone is on the upper side and plastocyanin is on the lower side. So the plastoquinone is located towards the outer membrane. Now this plastoquinone will also act as the proton carrier. Okay, so there are electron carrier as well as proton carrier. Electron carrier and proton carrier both are present here. So along with the electron carrier, we will also have proton carrier. What are proton carriers? They will also need protons to carry with them. Okay, so protons carrier can also transport the electron or carry the electron, but they will also need some protons together with the along with the electrons. Okay, so the protons carrier will carry an electron as well as proton. Okay, so this plastoquinone will carry an electron along with a proton. Okay, it will not carry an electron alone. So where will this get the proton? Since it is located towards the outer membrane, it will get the proton which was located outside. So you can see this proton. Let me zoom in. You can see in red color we have the proton located outside in the stroma. So this proton will also be carried by the plastoquinone along with the electron that was given by the photosystem 2. Okay, so it will carry both electron and proton, and then it will pass through the cytochrome complex, and then what will happen? The electron will be transferred to the photosystem 1, but the proton will be discarded into the lumen. So you can see this is the protons are being discarded in the lumen, because protons do not need to be transferred to the PS1. Okay, the photosystem 1 only needs electron, not proton. So electron will be transferred to the photosystem 1, but the proton has no use, so it will be discarded into the lumen of the thylakoid. Okay, so this is another reason why the proton concentration will increase in the lumen. So there are two ways we have seen in which by which the proton concentration is increasing in the lumen. First, during the splitting of water. Okay, it is taking place towards the inside, so all the protons which are released during the splitting of water are getting accumulated in the lumen. Second reason, the proton carriers are carrying the protons along with the electrons. They are transferring the electron to the PS1 and discarding the protons in the lumen of the thylakoid. So both these reasons contribute to a high concentration of protons inside the lumen of the thylakoid. Okay, then when the protons are being taken up from the stroma and being discarded inside the lumen, then it means the concentration of proton in the stroma will be decreasing. Okay, so the concentration of proton in the stroma is decreasing and the concentration is increasing in the lumen. Okay, now there is another way in which the concentration of proton is decreasing in the stroma. We know that in the stroma the reduction of NADP plus will be taking place, right? So the NADP plus requires a proton to get reduced. So where will it get the proton from? From the stroma. So it will take up the proton from the stroma to get reduced to NADPH. So this way also the proton concentration in the stroma will be decreasing. Okay. So by two ways the concentration of proton is decreasing in the stroma, and by two ways the concentration of proton is increasing in the lumen. Okay. So this I had to explain. I hope this is clear, how the proton concentration is increasing in the lumen and decreasing in the stroma. So let's read it now. The chemiosmotic hypothesis explains the mechanism of ATP synthesis. Okay, by studying the chemiosmotic hypothesis, we will understand how ATP is synthesized. Till now, we have not discussed how ATP is synthesized. We have just discussed how the proton gradient will develop. It will involve development of proton gradient across the thylakoid membrane. We have seen. Okay, accumulation of the protons in the lumen. The protons will get accumulated in the lumen. So how will the protons get accumulated in the lumen? By different ways. First is splitting of water on the inner side of the membrane, because PS2, the splitting of water is associated with PS2 and PS2 is associated with the inner membrane. Therefore, the splitting of water on the inner side of the membrane will lead to the release of protons inside the membrane, that is in the lumen. So this way the proton concentration is increasing in the lumen. Then second reason is that the electron is taken up by the electron acceptor, and then it is getting transferred to a proton carrier which will also carry a proton along with the electron, and then it will discard the proton in the lumen of the thylakoid. So this is why the proton concentration will also increase in the lumen. This is the diagram. This is the figure that we have already discussed, how the proton is being discarded in the lumen by the proton carrier. Then what will happen on the stroma side? On the stroma side, we have NADP reductase enzyme which will catalyze the reduction of NADP plus to NADPH and H plus. Okay, so the proton from the stroma will be taken up by the NADP plus. So this way the concentration of proton will decrease in the stroma. Okay, so in general what is happening in the lumen, the proton concentration is increasing, and when the concentration of proton is increasing, the pH will decrease. Okay, because pH is the minus log of H plus ion concentration. So when H plus ion concentration is increasing, the pH will decrease. Okay. Now, this is only half part of the chemiosmotic hypothesis. This has, till now we have only discussed how the proton gradient will develop. Okay, gradient means difference in the level. So now we have a high concentration of protons in the lumen and a very low concentration of proton in the stroma. So now there is a huge difference between the levels of protons in the lumen and the stroma. Okay, so this way gradient has developed. Now the energy will be released only when this gradient will be broken. So now the next step is the breakdown of this gradient. Okay, and this is the most important step, because only after the breakdown of the proton gradient the energy will be released, which will lead to the formation of ATP. Okay, so the next thing is breakdown of the proton gradient. So this is the thylakoid membrane. Now we know that we have a very high concentration of proton in the lumen of the thylakoid and very less concentration of proton outside, that is in the stroma. Okay, this is the stroma side. And inside we have lumen. So the concentration of proton is very high in the lumen and very low in the stroma. Now one thing that you have to remember, that when the proton was entering into the lumen to develop the proton gradient, that was the active transport. Okay, that was the active transport of the proton. Now the protons will try to get back, to move out of the membrane towards the stroma. Okay, so these protons will start moving out of the stroma, but this time they will be moving from a high concentration to low concentration. So this will be the passive transport of the protons. Okay, so during the formation of during the development of the proton gradient, the transport of proton was active transport, but during the breakdown of the proton gradient, the transport of proton will be passive transport. This you have to remember. Okay, so during the breakdown of proton gradient, what will happen? Movement of proton from the lumen to the stroma through the transmembrane channel of CF0 of the ATP synthase. Okay, so they will need some passage to move. Okay, they cannot move out from any place. Here in the membrane, we will have ATP synthase enzyme, which will have two components. One CF0 and CF1. Okay, CF0 and CF1, C for chloroplast. So CF0 and CF1 are the components of the ATP synthase enzyme. Now let's get back to the previous figure, the NCERT figure, to understand this. Here you can see we have ATP synthase enzyme here on the lower side of the membrane. You can see that ATP synthase enzyme has two parts, F0 part and F1 part. F0 part is embedded in the membrane, this part, and F1 part is located towards outside, that is towards the stroma side. Okay, so this long part, elongated part is the F0 part or F0 part, and this circular part is the F1 part. Okay, F0 part is embedded in the membrane and F1 part is protruding towards the stroma. Okay, so these two different parts have different roles to play. Okay, F0 part will help in the passive transport of proton from inside to outside. Okay, from the lumen towards the stroma. Okay, F0 part will help in the passive transport of the protons from lumen to the stroma. And what will happen to the F1? When the protons will move passively from the lumen towards the stroma, then F1 part will undergo a conformational change, which will lead to the generation of energy. Okay, and this energy will lead to the formation of ATP. Got it? So CF0 part of the ATP synthase enzyme will carry out the facilitated diffusion of the H plus or the proton across the membrane, and CF1 part, that is protruding towards the stroma, will undergo a conformational change due to the passive transport of proton from the lumen to the stroma. And because see, this is CF0 and this is CF1, right? CF0 will help in the passage of proton from the inside to outside. When the protons will be passed, then CF1 will undergo a conformational change, because this passage of proton will lead to the development of an energy or force known as proton motive force. Okay, proton motive force. So an energy will be generated which will lead to the formation of ATP. Okay, so F1 part will undergo a conformational change due to energy produced by the passive transport of proton, and it will lead to the formation of several molecules of ATP. Okay, so ADP and inorganic phosphate will come together. They will come together and they will lead to the formation of ATP. Okay, so this is known as phosphorylation, addition of phosphate to ADP. Okay, so this was about the chemiosmotic hypothesis. Now we have seen how ATP is formed. NADPH was already formed, that was simple. Okay, so what are the requirements of chemiosmosis? A membrane is required, that is a thylakoid membrane. We will have inner membrane and outer membrane. A proton pump that will help in the active transport of protons from the stoma to the lumen. Okay, so during the gradient development, the transport was active transport. Then a proton gradient will be formed, will be developed. Okay, that means higher concentration in the lumen and a lower concentration in the stoma. And then an ATP synthase enzyme is also important. Without this enzyme, ATP will not be formed. Okay, ATP synthase enzyme will have two parts, CF0 part and CF1 part. CF0 part will help in the passive transport of proton from inside to outside, and CF1 part will undergo a conformational change due to the energy produced by the passive transport. Okay, and then this conformational change in CF1 will lead to the addition of ADP and inorganic phosphate, leading to the formation of ATP. Okay, so now ATP and NADPH both are formed. We have seen how ATP and NADPH are formed during the light reaction. Now what is the use of ATP and NADPH? So these ATP and NADPH that are the products of the light reaction. These are the products of the light reaction will help in the biosynthetic phase. Okay, so they will drive the biosynthetic phase. Biosynthetic phase means the synthesis of sugar, that is glucose. So the synthesis of the food, that is glucose, will be driven by the products of the light reaction, that is ATP and NADPH. Okay, and this phase is known as biosynthetic phase or dark reaction. So this process of the synthesis of sugar or synthesis of glucose will come under the dark reaction. Okay, the formation of ATP and NADPH will come under the light reaction, and the formation of sugar will come under the dark reaction. Okay. Now this dark reaction consists of two different types of cycles, C3 cycle and C4 cycle. So here we have to study about two types of cycles, C3 cycle and C4 cycle. They will result in the formation of glucose, in the formation of sugar. Okay, C3 cycle is also known as Calvin cycle, and C4 cycle is also known as Hatch and Slack cycle. Hatch and Slack cycle. So now we will be studying about these two cycles, Calvin cycle and Hatch and Slack cycle. Okay, so Calvin cycle and Hatch and Slack pathway both are part of biosynthetic phase, right? And we know that during the biosynthetic phase, the glucose will be formed. So they will lead to the formation of glucose. The formula for glucose is C6H12O6. Right? Glucose formation will be common to Calvin cycle and Hatch and Slack pathway. Now in glucose molecule, how many carbon atoms are there? 6 carbon atoms. C6H12O6. So 6 carbon atoms are required. Where will we get these carbons from? We will get these carbons from the atmosphere. In the atmosphere, these carbons are present in the form of carbon dioxide. Okay, so initially the carbons are free in the atmosphere in the form of carbon dioxide. Okay, so many carbon dioxides will be there in the atmosphere. So these carbon dioxide molecules will be fixed for the formation of glucose. Okay, for forming one glucose molecule, we will need at least 6, not at least, exactly 6 carbons. Okay, so 6 carbon dioxides will be fixed to form one glucose molecule. So the step is known as carbon dioxide fixation. Carbon dioxide fixation. So this step, carbon dioxide fixation, will be common to Calvin cycle and Hatch and Slack pathway both. Okay. So what will be the main difference between the Calvin cycle and Hatch-and-Slack pathway? The main difference will be in the carbon dioxide acceptor molecule and the product that will be formed after carbon dioxide fixation. For example, let's write it down here. C3 pathway. We know that the Calvin cycle is also known as C3 pathway. Okay, it is also known as C3 pathway or C3 cycle. Whereas Hatch-and-Slack pathway is also known as C4 pathway. C4 pathway. Okay, C4 pathway is another name of Hatch-and-Slack pathway. Why they are known as C3 and C4 pathway, that we will see. In case of C3 pathway, that is Calvin cycle, there is an acceptor molecule containing 5 carbons. Okay, so we have 5 carbon molecule. Okay, you can see 1, 2, 3, 4, 5. 5 carbon molecule, that is known as RUBP. RUBP. And this molecule accepts the carbon. Okay, we know that the carbon dioxide from the atmosphere has to be fixed. So where will it get fixed? It will be fixed to some molecule. Okay, and that molecule will act as the carbon dioxide acceptor molecule. So in case of Calvin cycle, that acceptor molecule will be a 5 carbon molecule known as RUBP. Whereas in case of Hatch-and-Slack pathway or C4 pathway, the molecule will be a 3 carbon molecule. Okay, 3 carbon molecule known as phosphoenol pyramide, PEP. In short, we write PEP, phosphoenol pyramide. So in C3 pathway, the carbon dioxide acceptor molecule is a 5 carbon molecule called RUBP, and in case of C4 pathway, the carbon dioxide acceptor molecule will be a 3 carbon molecule, that is phosphoenol pyramide, that is PEP. Okay. So they will accept the carbon dioxide, and the carbon dioxide has 1 carbon. So RUBP was already having 5 carbons. Now it will have 1 more carbon of the carbon dioxide. So what will happen? 5 carbons, 1, 2, 3, 4, 5 of RUBP plus 1 carbon of carbon dioxide. How many carbons will be there? 6 carbons. 1, 2, 3, 4, 5, 6. So after the addition of carbon dioxide to the RUBP, we will have a 6 carbon molecule. But this 6 carbon molecule will be unstable. Therefore, it will break into 2 parts. Okay. You have to understand this carefully. So this 6 carbon molecule will break into 2 molecules having 3 carbons each. So 3 carbon molecule here and 3 carbon molecules here. And these molecules are known as phosphoglyceric acid or PGA, 3-PGA. Okay. So what is the stable product that is formed after accepting carbon dioxide? It is a 3 carbon molecule known as phosphoglyceric acid, PGA. Okay. In case of C4 pathway or Hatch-and-Slack pathway, the carbon dioxide acceptor molecule was a 3 carbon molecule, PEP, and then to this 3 carbon molecule, 1 more carbon will be added, that is from carbon dioxide. As a result, 4 carbon molecule will be formed. This is known as oxaloacetic acid. So in case of Hatch-and-Slack pathway or C4 pathway, the first product of carbon dioxide fixation will be oxaloacetic acid. Got it? Let's revise it again, because this is the main difference between the C3 and C4 pathway, which we often get confused. Okay, so in C3 pathway, that is also known as Calvin cycle, the carbon dioxide acceptor molecule will be a 5 carbon molecule known as RUBP. Okay, and this will accept the carbon dioxide present in the air. So carbon dioxide has 1 carbon. So 5 carbons of RUBP and 1 carbon of carbon dioxide will give 6 carbon molecule. But this 6 carbon molecule will be unstable and it will break into 2 molecules having 3 carbons each. So these 3 carbon molecules are known as phosphoglyceric acid, which is the first stable product of carbon dioxide fixation in case of Calvin cycle. Okay, so in Calvin cycle, the first product of carbon dioxide fixation is a 3 carbon molecule known as phosphoglyceric acid. This you have to remember. On the other hand, in Hatch-and-Slack pathway or C4 pathway, the carbon dioxide acceptor molecule was a 3 carbon molecule known as phosphoenol pyramide. Then 1 carbon of carbon dioxide was added to it. Then we got 4 carbon molecule known as oxaloacetic acid, which was the first product of the carbon dioxide fixation in C4 cycle. Okay, therefore they are known as C3 and C4 pathway. Why C3 pathway? Because the first carbon dioxide fixation product is a 3 carbon molecule, phosphoglyceric acid. And why that is known as C4 pathway? Because the first carbon dioxide fixation product is a 4 carbon molecule known as oxaloacetic acid. So this is the main difference that you have to keep in mind. Now let's study about the Calvin cycle first. It occurs in all the photosynthetic plants. So the Calvin cycle will be present in all the photosynthetic plants. We will be studying about two types of plants, C3 plants and C4 plants. Okay, in C3 plants, Calvin cycle will be operating only, and in C4 plants, the Hatch and Slack pathway will also be operating along with the Calvin cycle. So the Calvin cycle has to be present whether it is C3 plant or C4 plant. But the Hatch and Slack pathway or C4 pathway will be operating only in the C4 plants, not in the C3 plants. Okay. Now it was observed by the scientists. Calvin cycle is known as Calvin cycle because of the name of the scientist. Okay, the scientist was Calvin and his co-workers. Okay, so it is named after the name of the scientist. So the scientists observed that this pathway, C3 pathway, operates in a cyclic manner. Okay, that means the same pathway is repeated many times. So we have to study that cyclic pathway in order to make the pathway easy, make the understanding easier. We study the cycle under 3 different stages. Okay, so the first stage of the C3 pathway is carboxylation. What is carboxylation? Simply it means addition of carbon. Carboxylation means addition of carbon dioxide. So this is the most crucial step. We know carbon dioxide fixation or carbon dioxide addition is the most important step. So in this step, what is done? The carbon dioxide is added to RuBP because we are talking about the Calvin cycle, and in Calvin cycle the acceptor molecule was RuBP, the 5 carbon molecule, right? So it involves fixation of carbon dioxide into a stable organic intermediate. And what is that stable organic intermediate? 3 carbon molecule, that is phosphoglyceric acid, right? This is an intermediate. This is not the final product because the final product will be the glucose, not PGA. Okay. This is the most crucial step of the Calvin cycle because without fixing carbon dioxide, the process will not move ahead. Okay. Then in this step, carbon dioxide is utilized for carboxylation of RuBP. We have already discussed carbon dioxide will be added to RuBP. This is known as carboxylation of RuBP, right? And this reaction is catalyzed by the enzyme known as RuBP carboxylase enzyme. Why carboxylase? Carboxylase means it will add carbon dioxide, okay? But this enzyme can also add oxygen. So we can say that it does not have only carboxylase activity, but it also has oxygenase activity. Therefore, it is also known as RuBP carboxylase oxygenase. And in short, we write it as Rubisco. Okay, Rubisco means RuBP carboxylase oxygenase because it has carboxylase activity as well as oxygenase activity. It can also add oxygen, but here it will add carbon dioxide, not oxygen. And this stage carboxylation will result in the formation of 2 molecules of 3 PGA. We have already discussed. After addition of carbon dioxide to the RuBP, 6 carbon molecule will be formed, which will be unstable. It will be broken down into 2 molecules having 3 carbons each. So these 3 carbon molecules are known as 3 PGA, 3 phosphoglyceric acid. Okay. So this was the first stage of the Calvin cycle. Now we come to the second stage or second step of the Calvin cycle, that is reduction. Now this reduction is not just one reaction but a series of reactions, okay? Series of reactions that will lead to the glucose formation. Now what will happen during the reduction? Let's focus here, okay? We have got 2 molecules of 3 phosphoglyceric acid after carboxylation, okay? This is one phosphoglyceric acid having 3 carbons. This is another phosphoglyceric acid having 3 carbons, okay? So we have 2 molecules having 3 carbons each. Now, one of the carbons will be taken to form glucose, okay? One of the carbons will be taken, and next other 5 carbons will move to the next stage to regenerate the RuBP, okay? Because RuBP should have 5 carbons. So one carbon will be taken up to form glucose and next 5 carbons will be moving to the next stage, okay? Then another cycle will take place, one more carbon will be taken and the 5 carbons will be moved to the next stage. Okay. Similarly, the cycle will be repeating. When we will get 5 carbons, then glucose molecule, when we will get 6 carbons, then glucose molecules will be formed. That means at least 6 cycles will be required to form one glucose molecule. Now, the importance of this step is that this step utilizes ATP and NADPH. Okay, so 2 molecules of ATP are required during reduction for phosphorylation and 2 molecules of NADPH are required for reduction per carbon dioxide molecule. So we have already discussed that the ATP and NADPH which are formed during the light reaction will be used up in the dark reaction. So it is here where they are used. Okay. So how many ATP are required during reduction? 2 ATP for one carbon. Okay, we know that one carbon will be taken in one cycle. Okay, so for one carbon, 2 ATP will be required and 2 NADPH will be required for reduction per carbon dioxide molecule fixed. For making one glucose molecule, we will need 6 carbons. So 6 cycles will be required, right? So that means 12 ATP will be required and 12 NADPH will be required during this reduction. Okay. So have you understood? One carbon taken, 5 carbons sent to the next stage. Again, one carbon taken, 5 carbons sent to the next stage. And this way we will get 6 carbons and eventually there will be formation of carbohydrate at the expense of ATP and NADPH. Without using ATP and NADPH, this step will not be completed. Okay. Fixation of 6 molecules of carbon dioxide and 6 turns of cycle are required for the formation of one glucose molecule, because one glucose molecule contains 6 carbons. Okay, and during one cycle we get only one carbon. So exactly 6 carbons will be required. So for 6 carbons, 6 cycles will also be required, 6 turns of cycles. So this should be clear to you. This was the second stage of the Calvin cycle. Now we come to the third and the last stage, that is regeneration. We have already discussed that after carboxylation, we get 2 molecules of 3 PGA. Okay, this is 3 PGA. This is 3 PGA. We get 2 molecules having 3 carbons each. So total we have 6 carbons. One of the carbon will be taken and the other 5 carbons will be sent to the next stage, that is regeneration, where these 5 carbons will again combine to form RUBP. Okay, and that RUBP will again start the new cycle. So it involves regeneration of the carbon dioxide acceptor molecule, that is RUBP. What is the full form of RUBP? Rubisco 1,5,1,5 bisphosphate. Okay, so this will be regenerated, and again during this step 1 ATP will be required for phosphorylation of RUBP. Okay, so phosphorylation to make RUBP. So 2 ATP were required during reduction and 1 ATP is required during regeneration. So total 3 ATP will be required during one cycle and total 2 NADPH will be required during one cycle. This is the cycle you can see. Here we have ribulose 1,5-bisphosphate, that is RUBP. Carbon dioxide from the atmosphere was taken and added to this RUBP. So this is known as carboxylation of RUBP. So this is the first stage, that is carboxylation. And this step or this reaction was catalyzed by the enzyme RUBP carboxylase. RUBP carboxylase, which is also known as Rubisco enzyme. Okay. As a result of carboxylation, what was formed? 3-phosphoglycerate or 3-phosphoglyceric acid, that is 3 PGA was formed. Okay. How many molecules of 3 PGA? 2 molecules. 2 molecules of 3 PGA were formed. Then these 2 molecules of 3 PGA move to the next step, that is reduction. So this is the reduction. During reduction, how many ATP were required? 2 ATP and 2 NADPH were required. Okay, 2 molecules of ATP and 2 molecules of NADPH were required. So ATP was converted to ADP and NADPH was converted to NADP plus. Okay. As a result of reduction, we got triose phosphate, which were triose phosphate is the same thing, the 3 PGA which was move to the carbon from this was taken to form sucrose, starch, etc. Okay, to form the carbohydrate. Then the next carbons, the other carbons which were left after receiving one carbon were moved to the next stage, that is regeneration. During this stage, one ATP was used again. It was converted to ADP and the RUBP was regenerated to start a new cycle. Okay, so this is the complete cycle: carboxylation, reduction and regeneration. The important thing that you have to remember is during the reduction, for reduction 2 ATP and 2 NADPH are required, and during regeneration 1 ATP are required. But this is only for one cycle. Okay, for fixation of one carbon dioxide. For making a glucose molecule, we need 6 carbons. Okay, so we need 6 turns of cycle. So everything will be multiplied by 6. So for every carbon dioxide molecule that is entering the Calvin cycle, 3 molecules of ATP are required, 2 during reduction and 1 during regeneration, and 2 molecules of NADPH are required during the reduction. So for one glucose molecule, 6 carbon dioxides have to be fixed. So 6 into 3 ATP are required, that is 18 ATP, and 6 into 2 NADPH are required, that is 12 NADPH. 6 into 2 NADPH. Okay, so 18 ATP and 12 NADPH are required for the formation of one glucose molecule. So that you have to remember. So this was about the C3 pathway or the Calvin cycle. Now we come to the C4 pathway or Hatch and Slack pathway. Hatch and Slack pathway. Okay. Now this Hatch and Slack pathway will not be common to all types of plants. It will be operating only in some special type of plants known as C4 plants, which are adapted to dry and tropical regions. Okay, these plants lack photorespiration. What is photorespiration? It is a wasteful process. Okay, photorespiration is a wasteful process. There is no gain in photorespiration, only loss. Okay. So C4 plants lack photorespiration. That means the loss will not be there in C4 plants. So the C4 plants will avoid the loss. Therefore, they will have higher productivity. The C4 plants will have higher productivity than the C3 plants. Okay, this is important. The examples of C4 plants are sugarcane. Examples also are important: sugarcane, maize, sorghum. These are the examples of C4 plants. Okay. Now, the speciality about the C4 plants is that they have a special type of anatomy known as Kranz anatomy. What is Kranz anatomy? In the leaves of the C4 plants, we have the vascular bundles, right? So, suppose these are the vascular bundles. Now, around these vascular bundles, there will be some large sized cells known as bundle sheath cells, okay, which are present only in the C4 plants, not in the C3 plants. Okay, so bundle sheath are present in the C4 plants around the vascular bundles, and there is not just single layer of bundle sheath cells but several layers of bundle sheath cells are there. These are large sized cells around the vascular bundles. They have a large number of chloroplasts. Obviously, large number of chloroplasts means more photosynthesis will take place because chloroplasts take part in the photosynthesis. Then they have thick walls which are impervious to gaseous exchange. Okay, so gaseous exchange does not take place. And there are no intercellular spaces. Okay, these are the special features of the Kranz anatomy. This type of anatomy, the leaves which have this type of anatomy are known to have Kranz anatomy. And Kranz anatomy is the feature of C4 plants only, not C3 plants. Now let's see how the glucose will be formed in case of C4 plants. In C3 plants, it was simple. In C4 plants, you can see the figure here. You can see two cells are shown. Okay, two compartments are shown here. This is upper compartment, this is lower compartment. Okay, and there is plasmodesmatal connection between these two. Okay, so the contents between these two can be shared, right? So what are these two compartments? The first compartment, the upper one, is the mesophyll cell. It represents the mesophyll cells, and the lower compartment shows you the bundle sheath cells. Bundle sheath cells which are present only in the C4 plants, not in the C3 plants. Okay, in C3 plants only mesophyll cells were there. Okay, we have studied Calvin cycle that takes place in the mesophyll cells of the C3 plants, but here the Calvin cycle will not take place in the mesophyll cells. It will take place in the bundle sheath cells. This is one of the difference. So what will happen in the mesophyll cells of the C4 plants? Let's see. Here we were discussing the main difference between the C3 and C4 cycle. There we had discussed that in C3 cycle, the carbon dioxide acceptor molecule was a 5-carbon molecule RUPP, whereas in case of C4 plants, the carbon dioxide acceptor molecule was a 3-carbon molecule. 3-carbon molecule, that is phosphoenol pyravate. In short, it is written as PEP. So here you can see we have phosphoenol pyravate. Okay, is it clear? Phosphoenol pyravate. Then atmospheric carbon dioxide was taken in the form of bicarbonate. The atmospheric carbon dioxide was taken, it was converted to bicarbonate, and then this bicarbonate was added to the phosphoenol pyravate. So this way carbon dioxide fixation took place, and what was formed? 3 carbons of the phosphoenol pyravate and 1 carbon of the carbon dioxide, 4 carbons. So we have got a 4-carbon molecule or a 4-carbon acid known as oxaloacetic acid. Okay, it is taking place in the mesophyll cells of the C4 plants. So what happened? Carbon dioxide from the atmosphere was converted into bicarbonate and taken up into the mesophyll cell. Then this carbon dioxide was added to a 3-carbon molecule, phosphoenol pyravate. Then we got 4-carbon molecule, that is oxaloacetic acid. This is the first carbon dioxide fixation product of in case of C4 pathway. Okay. Now this C4 acid, that is oxaloacetic acid, which was formed in the mesophyll cells of the C4 plants, will be transported to the bundle sheath cells through the plasmodesmata. Okay, so these two mesophyll cells and bundle sheath cells are connected through the plasmodesmata. So this oxaloacetic acid was formed in the mesophyll cells and it was transported to the bundle sheath cells of the C4 plants. Now what will happen in the bundle sheath cell? Let's see. Here decarboxylation of the oxaloacetic acid will take place. Okay, oxaloacetic acid reached to the bundle sheath cell, then decarboxylation took place. Decarboxylation, that means removal of the carbon dioxide. So from this 4-carbon molecule, oxaloacetic acid was a 4-carbon molecule, carbon dioxide has been removed. That means 1 carbon has been removed. So what will be left? 3 carbons will be left. Okay, so we will get a C3 acid. After the removal of carbon dioxide from oxaloacetic acid, 3-carbon acid will be formed, which will be again transported back to the mesophyll cell. Okay, so oxaloacetic acid had come to the bundle sheath cell only for decarboxylation, right? So this C3 cycle will be transported back to the mesophyll cell, and then this C3 cycle will again get converted into the phosphoenol pyravate, which will start a new cycle in the mesophyll cell. But what will happen to the carbon dioxide that has been removed from the oxaloacetic acid in the bundle sheath cell? So this carbon dioxide will be fixed by the Calvin cycle. Okay, so this carbon dioxide will enter the Calvin cycle taking place in the bundle sheath cell. Okay, so here the Calvin cycle is taking place. So what we have seen that in case of the C3 pathway, the carbon dioxide fixation, the carbon dioxide was taken directly from the atmosphere and fixed during the Calvin cycle. But here in the C4 plants, the carbon dioxide is not taken directly from the atmosphere. Several things had happened in the mesophyll cells. Then the oxaloacetic acid was formed. That oxaloacetic acid was transported to the bundle sheath cell. The carbon dioxide was removed from it, and then this carbon dioxide was used in the Calvin cycle. Okay, so in this C4 pathway, this C4 pathway is quite complicated as compared to the C3 pathway. Okay, there Calvin cycle had taken place simply, but here for Calvin cycle, so many things had happened before it. Okay, so I have tried to explain it in the best way. If you have not understood, you can repeat the video again. Okay. So the important thing is that the Calvin cycle in case of C3 pathway was taking place in the mesophyll cells. Okay, in C3 pathway, the Calvin cycle was taking place in the mesophyll cells because the C3 plants had only mesophyll cells. They did not have bundle sheath cells. Okay. And the carbon dioxide was taken directly from the atmosphere. But in case of C4 plants, the Calvin cycle take place in the bundle sheath cells. So in mesophyll cells, the Calvin cycle does not take place. It takes place in the bundle sheath cell. Also, this time the carbon dioxide that is used during the Calvin cycle has been taken from the oxaloacetic acid which was formed in the mesophyll cells. Okay. So I hope you have understood the C3 and C4 pathway. Now we have come to the last topic of this chapter. This is a small topic, photorespiration. We have already discussed about the photorespiration, that it is a wasteful process, right? It is a wasteful process. There is no gain in this process, only loss. Let's see how. We know that the C4 plants lack photorespiration, but C3 plants do not lack photorespiration. So in C3 plants, photorespiration does take place, right? What happens in C3 plants? We know that the enzyme, Rubisco enzyme, here we have the Rubisco enzyme, full form is ribulose 1,5-bisphosphate carboxylase oxygenase, right? So this enzyme has carboxylase activity, that means it can add carbon dioxide, as well as oxygenase activity, it can add oxygen as well, right? So sometimes in C3 plants, it binds with oxygen rather than carbon dioxide, right? So what will happen if oxygen will bind to it? It will decrease the carbon dioxide fixation, okay? Because RUBP has to bind with carbon dioxide to fix the carbon dioxide and convert it into glucose. But here it is not binding with carbon dioxide, instead it is binding with oxygen. So carbon dioxide fixation will be decreased. So there will be lower glucose production. So instead of being converted to two molecules of 3-PGA, during the carboxylation, what is formed? Two molecules of 3-PGA are formed, right? So instead of being converted to two molecules of 3-PGA, RUBP will form one molecule of PGA and one of phosphoglycolate. Okay, so we know that the RUBP molecule is a 5-carbon molecule, 1, 2, 3, 4, 5. One carbon from carbon dioxide will be taken, so we will have 6 carbons. Okay, 6-carbon molecule. Then this 6-carbon molecule will be broken down into two molecules of 3 carbons each, because the 6-carbon molecule is an unstable molecule, right? So two molecules of 3-PGA have to form. But here this carbon dioxide is not being taken, oxygen is being taken. So where will this carbon come from? Only 5 carbons will be there of RUBP, right? So this will give one molecule having 3 carbons, that is PGA, and other molecule having only 2 carbons, that is phosphoglycolate. Phosphoglycolate. So phosphoglycolate is a 2-carbon molecule and it has no use in photosynthesis. Okay, it has no use in photosynthesis. So obviously it is a waste product here, right? Because we have no use of phosphoglycolate. So as a result, in C3 plants, when the Rubisco binds with oxygen, then no sugar formation will take place because carbon dioxide fixation has not taken place. No ATP will be produced, okay? No ATP or NADPH. And there will be release of carbon dioxide with utilization of ATP, okay? So during Calvin cycle, what should happen? Carbon dioxide should be fixed. So here carbon dioxide is not being fixed. Instead, carbon dioxide is being released. Okay, so it is a wasteful process. Also, the ATP is utilized here. So nothing is gained, but ATP is utilized. So we have loss of ATP. Okay, so gain is 0, but loss is more, right? But in C4 plants, photorespiration is not seen. Okay, it lacks photorespiration. Photorespiration. How does C4 plant lack photorespiration? Because it has a mechanism to increase the concentration of carbon dioxide. Okay, it has the mechanism to increase the intracellular carbon dioxide concentration. How? We know that in the C4 plants we have one mesophyll cell, then it is connected with the bundle sheath cell. Okay, mesophyll cells of C4 plants are connected with the bundle sheath cells. In the mesophyll cells, what is formed? 4-carbon molecule, that is oxaloacetic, is formed. This oxaloacetic acid is transported to the bundle sheath cell. Then this oxaloacetic acid is broken down. Okay, here decarboxylation takes place. So oxaloacetic acid, from the oxaloacetic acid, carbon dioxide is removed. Right. So this process is taking place continuously. That means continuously the concentration of carbon dioxide is increasing in the bundle sheath cells. Okay. Now here we should know one fact about the ribisco enzyme. Ribisco enzyme. We know that it has an affinity for carbon dioxide as well as oxygen. It can bind with carbon dioxide as well as oxygen. Right, but it has a higher affinity for carbon dioxide. Suppose the concentration of carbon dioxide is more, then it will bind with carbon dioxide. If the concentration of oxygen is more, then it will bind with oxygen. But what will happen if the concentration of carbon dioxide and oxygen both are equal? In that case, it will bind with carbon dioxide. So it has high affinity for carbon dioxide. Affinity for carbon dioxide when the carbon dioxide and oxygen both are present in the same amount, or in same concentration. So if the concentration of carbon dioxide will be more, then it will bind with carbon dioxide. If the concentration of oxygen will be more, it will bind with oxygen. But if the concentration of carbon dioxide and oxygen is same, then it will bind with carbon dioxide because it will have higher affinity for carbon dioxide. So this also you should know. But here what we are discussing, in C4 plants, the oxaloacetic acid is giving carbon dioxide continuously in the bundle sheath cells. Okay, decarboxylation of oxaloacetic is taking place continuously. So there is a continued increase in the concentration of carbon dioxide in the bundle sheath cell. So there will never be a time when the concentration of carbon dioxide and oxygen will be same. Always the concentration of carbon dioxide will be more than the concentration of oxygen, only in case of C4 plants. In C3 plants, there is no such mechanism. Okay. So here the ribisco will always bind to the carbon dioxide. It will never bind to the oxygen. Therefore, photorespiration will never take place in the C4 plants. Got it? So this will result in higher productivity and yield. Higher productivity and yield in case of C4 plants. Okay, so which plant is better? The mechanism of which plant is better? C4 plants or C3 plants? C4 plants, because in C3 plants photorespiration can take place, which is a wasteful process and that can decrease the productivity. But in case of C4 plants, the mechanism is so specialized that there will never be a decreased concentration of carbon dioxide, and ribisco will always bind to the carbon dioxide, leading to higher productivity and higher yield. Okay, so C4 plant is better than the C3 plants. And this C4 plant also has tolerance for higher temperature. Therefore, these plants are adapted for higher temperature areas. Okay, for dry areas and tropical areas. Okay, so this was one of the differences between C3 and C4 plants again. Now we have completed this topic. I hope you have understood the photosynthesis clearly. If you still have any doubt, then you are always free to mention your doubts in the comment section. I will be replying to your doubts. Even if you want me to explain some topic again, I can do that also. Okay, so we will be back with another session soon. Till then, keep learning, keep scoring high. Thank you so much.